Genes of you and me, genes of you and you're all made of DNA. We're all made of the same chemical DNA. We're all made of DNA. We're all made of the same chemical.
Hello, and welcome to the 16th episode of Discovering New Advances, or DNA, a podcast that keeps you up to date about the scientific world in easy-to-understand language. So whether you're a student in middle school or a grandparent, you'll easily be able to follow along. I'm Kira, and on this episode, I will give an introduction lesson about fluorescence in situ hybridization, also known as FISH analysis for short. And it's not as complicated as it sounds, I promise. The following episodes will either keep you updated on genetic advances in the scientific world, teach more lessons on genetics, or feature a debate about controversial bioethical issues our society are facing today. So if you're already familiar with the basics of interphase fluorescence in situ hybridization analysis, you can skip to the following episodes about news, genetic advances happening today, and how it is affecting our world. For more information, please visit dnapodcast.com or email in at info@dnapodcast.com. So on this lesson, as I previewed earlier, we're going to learn about fluorescence in situ hybridization analysis, known as FISH analysis for short. FISH is used to find specific DNA sequences on chromosomes. It does this by physically illuminating DNA sequences so the cytogeneticist can see it. So it's kind of like a neon spot on an image. It was developed by biomedical researchers in the early 1980s, so not that old, just like many of the genetic technologies we have today. So let's find out how this actually works. Chromosomes are denatured on a slide, and a DNA probe sticks to a particular segment of DNA. Now, the DNA probe has a DNA sequence of its own and attaches it to complementary sequences of DNA. So that's why it's that identifier that it has a specific sequence that scientists decide they want to find in the cell, and it complements the gene that they're actually looking for. So the DNA probe also has this fluorescent dye on it, which makes the segment of DNA illuminate to clearly be visible, kind of as I said before, as a neon spot. And there are different types of FISH probes, which all highlight different unique sequences, so you can really find whatever you're looking for. So these include whole chromosome painting and repetitive probes. So why would cytogeneticists need to identify specific sequences of DNA? Well, it can help them pinpoint where a specific gene falls on an individual's chromosome. So what makes FISH a flexible procedure is that it does not need to be performed on cells that are actively dividing. So I mentioned this a couple episodes ago when I previewed FISH, but karyotyping, you need to have a cell in the mitotic phase, but in FISH you don't. So this is a great advantage that it doesn't need to be dividing all the time, because in research you'd have to wait until a cell is dividing in order to do certain techniques on it. So this, you don't have to wait for it to divide. So another advantage is the ability to use different probes to highlight different parts of the cell, and it's not just genes. You can highlight different proteins, so that is really helpful to outline, certain organelles, and some really pretty pictures, beautiful pictures of cells are taken using the FISH analysis, because they can illuminate different parts of the cells, and that's what you're seeing in your textbooks probably. So to review all this information, FISH analysis, or fluorescent in situ hybridization, which is the official term, hybridization analysis, official term, is the method of identifying sequences of DNA. This is accomplished through DNA probes, which attach onto different DNA sequences depending on the sequence the DNA probe has. This allows cytogeneticists to see genes and to find where they are in the cells. Well, thanks for listening. I hope the lesson helped you out a little bit. You can further your education by visiting dnapodcast.com for more episodes, and this lesson, along with others, not only in audio format, but in a written format with visual aids to help you out a little bit more. There's also a contact form on there for easy access to send in feedback about the shows or any questions you might have. You can email us directly if that's easier for you. That's info@dnapodcast.com. If you're as obsessed with social media as I am, you can like us on Facebook and follow us on Twitter, both at /DNApodcast. Subscribing to us on iTunes is a great way to keep up with the show. It will let you know when new episodes are out and automatically download them for you. That way you don't have to keep checking back with the site. So all you have to do is search DNA Podcast and we pop right up. If you have iTunes open, you might as well give us a review. That's the best way to have our podcast viewed by more people, and that way more people can benefit from it, just like yourself. You can find all the links I just mentioned, Facebook, Twitter, iTunes, and an RSS feed on the website dnapodcast.com. Thanks for listening, and join me next episode to learn and discover new advances in the world of genetics.